Welcome back, everyone. Next up, we have GASE Energy Inc. It trades on the OTC Pink sheets under the symbol GASE and recently signed an LOI to acquire Sanatio BioScience Inc., SBI, a modular therapeutics antiviral-anticancer platform which uses biologic components in tandem arrays that target specific cell types and tissues to enhance the natural antiviral activities within the cells. The company has a near-term focus on COVID, but the Sanatio platform can be configured to address new threats as well as strains of existing viruses. Please welcome its CEO, Judson Boothe, and CEO, Sean Martin. Welcome, gentlemen.
Thank you. Thank you. Proud to be here. And hopefully I can summarize it as best you just did. As mentioned, I'm the CEO of GASE Energy, which is a fully compliant OTC public company trading under the symbol GASE. I'm proud to be joining forces with Judson Boothe, who's with us today, who will be going over the formality of Sanatio Bio with the presentation. I will say that the focus of GASE in its transformation will be on a platform of biotech opportunities. I think we've all witnessed a pretty significant correction in biotech, and I think personally that leads to opportunity, which is why I'm so excited to be initially partnering with Judson and his very experienced team. So I will say that we're going to be on the lookout for other opportunities exactly like this, because I think with the correction, there's a myriad of things that we've been working with, not just with COVID, with cancer and other opportunities that are synergistic with what we're doing here today. So without any further ado, I'll put it in the hands of our very well-talented Judson Boothe to explain exactly what our initial acquisition is all about. So Judson, all yours.
Thanks, Sean. I'll spend the balance of the time today talking about Sanatio and what we're doing, what the company's about, and then we'll hold some time at the end for questions. Both Sean and I will be available to answer any questions that you may have. First of all, what is Sanatio? You've heard of the introduction and was mentioned in the press release on the merger intent. But Sanatio is a modular therapeutics company focused primarily right now on antiviral with a phase 2 plan for anti-cancer. It's a platform company. We are going to talk a little bit about what the platform is, but let me just describe it in the following way. We are targeting viruses and cancer because we can go after the specific genetic materials that those two things give off. In the case of viruses, we go after proteins. In the case of cancer, we go after some of the byproducts produced by the tumors. It's a platform company. It's not specific to just one virus or one cancer type. We believe the platform can be targeted and configured to rapidly adapt to different targets. We'll talk a little bit more about that in some detail. It's a modular technology in that it consists of several different things in the therapeutic that each have their own role. And the novelty is in the combination of the modules. So it's not a one-shot therapeutic that only does one thing. It has multiple features. Each feature provides a piece of the puzzle, and the things together work effectively to go after, in the first case of the viral threats, we'll walk through how they work. It is rapidly adaptable. So unlike a vaccine, which takes in recent times, it's not taken as long as it has in the past. But in normal times, vaccines take quite a long time to develop. Our therapeutic being an antiviral, as soon as you can sequence the DNA of a new target, we can rapidly change our therapeutic to target that candidate. It's very, very quick. It's very easy to adapt to. And the ability to identify the targets is very common. We've already worked on all the different deviations of COVID now, and they've all been sequenced, and so they can all be targeted as an example. It's also rapidly adaptable. All of the production techniques that we use and the materials we use are readily available. It doesn't require a unique manufacturing process to make. It should be readily adaptable and configurable to many different labs and many different locations, so it's easily produced. We'll talk a little bit more about the problem that we're solving. I'm not going to get too much into this. We all know what COVID has done and what COVID has looked like. It's also interesting to note that this is an antiviral that we're going after. It's not a vaccine. It does not create immunity. We are going after candidates that have live viral threats, and we are going to eliminate the threat to the patient, therefore minimizing the risk of hospitalization or ultimately really bad outcomes, such as even death in some cases. The problem that we're solving right now, we're using COVID as a proof point. We've tested initially with some of the initial variations or mutations. We'll be testing further the more recent variations. We'll talk a little bit about Omicron and Omicron most recent mutation in a moment. But we're testing against all of the COVID strains as a proof point. We also have in our lens things like SARS, RSV, MERS, influenza, or any other new viruses that crop up that we can go after and target. So once we're configured against COVID and have proven that we can do that, we'll go after these other candidates as well. So it's not just a COVID solution. The toll that some of these things have taken are massive. We all know that, COVID's been the topic of conversation for the last 2 years, but pandemics are not new. You go way back in history, we've had events of these types all along our timeline, and we'll continue to have more. And so we're building a platform and a therapeutic module that can be tailored against not only the current threats, but also future threats. Again, DNA sequencing is very common. Once we can sequence the virus, we can target it. It's as simple as that. Let's talk a little bit about the new BA.2 strain of Omicron, which is the most recent mutation of the COVID-19 virus. Word is right now that we expect that this virus could be more transmissible. It could cause some of the more significant effects like the earlier strains did, such as Delta. It's not as mild as the initial Omicron strain has been. It also seems to be more resistant to the current vaccines, is what most of the studies are saying now. We're also believing that this could be the dominant variant worldwide very quickly. Right now, it accounts for less than 5% of the cases in the U.S. Just an example of these viruses will continue to migrate over time, and the vaccines will have to continue to regenerate themselves. We, as a therapeutic, would also have to continue to retarget ourselves to the Omicron BA.2 variant if that were to be the dominant variant. The easy part is that we are targeting the protein within the virus, which parts of that protein or most of that protein don't really change from mutation to mutation. Some parts do, some parts don't. If we can successfully target a non-mutated part of the virus, then we are equally as effective against any of these strains as we were the prior. So that's part of the novelty, is we target a non-mutating part of the genetic material on the virus. The therapeutic is a modular component which allows us to go after the certain things related to the specificity of the cells, as well as allowing us to get into the cells easily. So under normal circumstances, the cells don't want foreign materials to come into it. Getting through the cell membrane is not exactly easy. So part of what our therapeutic does is it allows access to the cell so that we can deliver the targeting material to where the virus is, which is inside the cell. We also can, with other modules on the design, we can target the specific cells. As an example, we can target mostly lung cells if we choose to by adding different modules to the therapeutic. In this case, in COVID, we're targeting lung cells. We're targeting access to those lung cells, and we're targeting a protein on the COVID-19 virus, the spike protein in particular. Sanatio is formed by a pretty seasoned team of people. I'll talk a little bit about that right now. And we also talk a little bit about what we've accomplished so far relative to IP and studies so far. The team consists of myself. I've been in the medical space for around 30 years, mostly with larger companies doing medical device, large commercial operations. The two scientists on the team are Peter, and we affectionately go by Cassia for Dr. Cassanathan. Peter is the primary designer and developer. Both are affiliated with Rutgers and have worked extensively in biologic sciences for a number of years. Frank Knettel is our financial advisor. He is helping us out with keeping our finances straight, and this forms the core team. We also have other advisors. We have other scientific advisors and partners. Some of our partners that we're working with, GD3 is one of our primary CROs. Biotech is a specialist working on the targeting mechanism of our therapeutic, one of the best in the industry at what he does, and that team. And then Southern Research is where we're conducting our cell studies, as well as we'll be doing our animal studies with Southern Research out of Birmingham, which is right near UAB in Birmingham, Alabama. We do have Joseph Sheridan on our team as a scientific advisor. Joseph is a senior scientist at Moderna currently. He has joined our team to help us with scientific advisement, and we rely on his guidance a lot in designing experiments and selecting CROs and things like that. So he's a core part of our advisory team. We also use a number of advisors that are part of the CROs that we work with. So GD3, for example, has a number of people that we've worked closely with that have contributed greatly to the production of the therapeutic. So where we're targeting right now, let's talk about the way this thing works. Each module has its own function. The biologic nature of this means that it is not a drug, it's actually a biologic. It will be filed as a BLA submission for use versus an NDA due to the biologic nature. All the modules are proven history and safety. So each module consists of things that are used or existing in the world today that have a proven safety background. So we're not creating new material that we worry about, things like toxicity and interactions with the human body. As I mentioned before, we use standard manufacturing practices. We believe they can be replicated globally and relatively cheaply. And the tissue-specific targeting is really trying to drive the therapeutic to where the infection is. So many drugs are really a blitzkrieg of the body, where you just blast the body with, holistically and try to make sure you get to where the virus is. We believe we can actually target the tissue where the virus is concentrated. So the module consists of cell targeting materials. Cell access and specificity is what we refer to it. If you look at the top right chart, that's probably the easiest way to describe it. Cell access and specificity. That's where we go after really specific cells, and we have materials in the therapeutic that allow us to get into that cell easily. We take our therapeutic across the cell membrane, getting inside the cell, as you see on the photo to the left. That's an example of the cell. The virus lives inside the cell. We have to get in there so we can target the virus. Viral targeting, this is a mechanism within the therapeutic that goes after only the target candidate. If it doesn't find that viral target or that specific protein on that virus, it will not target anything else. So it's a one-shot deal that it's looking for its specific target. If it doesn't find it, it really doesn't do anything, just gets absorbed by the body. And then the antiviral activity. So when our target is found and bound by the therapeutic, the natural antiviral activity of the body, or the enzymatic activity that tears apart the virus protein strand, goes into action. We work one of two ways. We can either bring that with us and provide our own activity, or we recruit within the body itself and use the mechanisms that are already there and enhance that. Both of which are in study groups, as you'll see the versions down below. Therapy 1 is where we are today, which is using the native endogenous antiviral activity. So we're not bringing in material with us. We're keeping it very simple and using what's already existing in the body. We can supplement that with other concepts of the therapeutic where we bring that with the modules. So where are we at timeline-wise? We have created materials. We have done our initial cell culture studies with infected cells in a high-throughput screening module. We are then now approaching development of optimization of those therapeutic options within cell culture, and then getting ready for our first round of animal testing, which will be in hamsters. Hamsters are the best test case right now for a COVID material because it has a high, the ACE2 receptors in a hamster are very similar to the humans, and that's the cell types that COVID tends to like. So very simply, we build the modular therapeutic, we formulate the prep, and then we test in cell culture and animal. That gets us to proof of concept through animal studies, which then we go into pre-IND work for our human trial plan. The good news about where we're going is we think this can be done pretty quickly. We don't think it'll take us much longer than about 16 weeks or so to get through this next phase of having animal study data to look at. The progress to date, I mentioned the initial study. We've also filed a patent application for the modular system with multiple modalities, both in virus and with some of the cancer work called out as well. Multiple modalities being multiple types of viruses that we've included. So that patent has been filed. Initial rounds of study compounds designed and formulated. We've figured out how to make them and hook them together into a therapeutic. We've ran initial cell culture study at Southern Research, and we've applied for a grant with NIH, or that grant application is in process. Mentioned before the next steps, optimization of the compounds and formulation, confirmation and selection. Cell culture study will choose the candidates to take forward into the hamster study, and then we'll do the initial animal model study with infected hamsters. Very briefly, I'll talk about the study that we did, and then I think we'll be at a point where we can open it up for questions. The cell culture work is done on an infected cell line, and we measure our effectiveness against that cell by taking down the cell, the amount of infection in the cell, and we want to make sure that the cell remains healthy. So we worry about both killing the virus and keeping the cell healthy. So we measure both toxicity and we measure the effectiveness of the antiviral. The curves on the left are the two candidates that we had that showed really well. And the number you're looking for is what's called that IC50. You'll see a number there in the left-hand side of those charts that says IC50. That's the time it takes, or the concentration it takes to kill 50% of the virus, basically. So it's at what point you have 50% effectiveness, and that's a measure of the effectiveness of the candidate compounds. You can see some comparison compounds over on the right. You've heard of remdesivir probably in the news. Down about 4 from the bottom, you'll see the remdesivir IC50 is 15. We had IC50s of 11 and 20, which are right in the ballpark of some of those compounds that are viewed as relatively effective. Now, remdesivir has a lot more complications. It has to be given by IV. It has to go in multiple rounds. It's very expensive. It's very time-consuming for the patient to go through the treatment. So there are reasons that it hasn't taken off as a treatment. We wouldn't have that same complication factor. But the good news is we're seeing high levels of activity, much like some of these other things that we've seen. And this is without optimization. We believe we can improve on these numbers with our next round of optimization. On the right-hand side, the toxicity charts, if you saw a problem, you would be seeing a downward trend in the data. So as concentrations went up left to right, you would see a downward trend in the cell energy, and therefore you would see toxicity effects. We actually saw no toxicity effects whatsoever, as we expected. These compounds are expected to be relatively non-toxic, and that's what's proven out to be the case. So that's our first cell culture study data. Again, summarizing where we're at, we're excited about the opportunity to work with GASE. We've done some initial work that we feel very confident about. We're ready to take our therapeutics up to the next level, which is a bigger round of studies and in through animal studies. And then we'll be looking at taking it forward from that, assuming we have viable candidates coming out of those rounds. I will flip it back to Sean if he has any other comments, and if not, we can open it up to Q&A if Sean has no additional comments.
No, Judson, I think you did a great job. Happy to field some questions from the audience.
All right. Thank you, gentlemen. We do have some for you. Michael Wanner wants to know, what's your stance on a reverse split?
So the cap table of GASE right now, let's go to OTC. You can find out the fully diluted, et cetera, on GASE. I do believe there is a possibility of a reverse stock split pending the merger with Sanatio. It's something we're going to discuss to be commensurate with a reasonable share count, commensurate with other OTC compliant companies. So it's something that we could do. We haven't made a decision on that, but it's something that we may have to look at in the future.
Okay, and same gentleman asks about what are your revenue projections for 2022 and 2023?
I'll take some of that. I think we'll still be in research phase in 22. 23 remains to be seen. It really is dependent on how fast we get through, any human trials that we do that are required to take it to availability. So right now, I don't expect any revenue in 22, and 23 is, we'll see based on how long the human trials take to get there, and our partnership opportunities. I expect we might be looking at partnering post-animal studies with an existing commercial entity, and that could drive that faster, and/or look at licensure or whatever in 23.
Okay, and talk a little bit about Sanatio. What is the current revenue, and how do you plan to acquire Sanatio? And if you're issuing shares, will they be preferred?
If that's for me, we will not be issuing preferred shares. It's a straight equity combination. And I also wanted to go back to your prior question regarding the stock split. And I've mentioned this before publicly that I'm the largest shareholder of GASE, and I'm all about preserving shareholder value here. I am willing to cancel a percentage of my GASE shares pursuant to the involvement in a potential reverse stock split. So let me just say that there. We will be issuing additional shares, however, for the split between GASE and Sanatio Bio, which is in our LOI.
Okay. And Irvin Reese asks a question about your public company. Are you acquisition minded? And if so, what do you look for exactly?
Well, I think Sanatio is kind of a good example. We are obviously an early stage investor. I've done this several times before. We are an early stage, let me say an investor in companies just like Sanatio. So I would like to stick in the oncology space, pre-revenue, not necessarily preclinical. We have looked at another company, and many of my followers know that very well, a company called Curadox. We have not turned down the opportunity with Curadox, which is an oncology company focused on chemotherapy. But, examples of that, and my followers know that, are ideas that we're looking at right now. As I mentioned, and we're all aware, the biotech industry has taken a big hit, but that presents opportunities for us. So we'll be canvassing many opportunities going forward.
Great. Brad Lucas wants you to talk about how you're capitalized and how you plan to raise some money to further the development of your technology.
Sure. Let me just start with that. I wanted to have Judson go over a little bit about our grant opportunities, which are non-dilutive. So maybe, Judd, you could give us a little update on what we're looking at there and our progress.
Yeah, so we've engaged a grant specialist to work with us on going after a number of non-dilutive government grants. Those would be, in 22, so we would use part of that as well as, equity funding to raise the capital to go through the next round. And then with combination with GASE, obviously that opens up the public markets opportunity as well. So those are the three avenues for revenue raise, or for investment raise.
Okay. Rata Trader asks, if you receive your grant, how much is it, and how far will it take you before you have to raise some funds?
There are different types of grants. There are phase 1, phase 2 grants. Phase 1 grants from the NIH are generally a few $100,000. Phase 2 are usually in the couple $1 million range. The first level wouldn't take us far enough to get through animal studies. Most likely, we're probably going to have to go either the bigger grant route or combination of grant and equity raise to go through the next phase of research. We think we're a few $1 million. We get through the animal studies, no problem. And that gets us to the next value creation point, which is solid animal data that says our therapeutic is effective against COVID. And then we can, we have some optionality at that point in what we do. We can go after proving different viruses with the same platform, or we can go after further developing the COVID platform into the early stages of getting ready for human studies, or we can do both depending on the the capitalization and the revenue and the investment that we have at that time. So we have some options after we get through this next round of animal studies.
Great. Winston Cooper asks, when you say you work on all variations of COVID, what types of studies and trials have been done?
We've only done one study on one of the early variants of COVID, but the way we work is we go after the spike protein on the virus itself. So the spike protein is a very, very, very long protein. So it's a very large protein, and we target a very small amount of it. So we probably target less than 1% of the protein length. The mutations are occurring all along that protein at different times, for all these different variations. But the whole protein itself doesn't mutate. It's only mutating in very small places. And we target more than one area. So as long as we have any one area that we're targeting that hasn't mutated, it will be fine to work against it. Now, we'll need to test all of those to make sure that the target areas we're going after are not part of the mutated protein. But when they sequence those proteins of the new variants, we know where we've targeted. They can sequence it, and we know where it's mutated, so we know empirically that it's not part of the same area where we targeted, so we know that it's going to work fine. It'd be unlikely to not work. But I would still say part of our testing plan would be to test against every variant just to validate that and make sure that it's still true. But scientifically, there's no reason it shouldn't work as long as a mutation didn't occur on the target area that we're targeting with our therapeutic.
And Roy Frazier wants to know, how much money have you raised in the past and at what valuation?
I lost part of the question. I heard how much, and then I lost it.
Sorry. How much money have you raised in the past and at what valuation?
This is for Sanatio or for GASE or for both?
Let's go for both.
Both.
Okay. Sanatio, we're currently self-funded to date. Only the founders have put in money, and we've done for less than $200,000 the work so far. So it's relatively low capitalization so far. Again, we just tried to get a candidate material into a cell culture study so we could see if it worked or not. Now that we have it, we're going after the next piece. At the valuation, I can't remember, Sean, do you know the valuation off the top of your head that we've been using?
I would say in the beginning it was about a $2.5 million valuation. Right now we'd be looking at about a $9 to $10 million pre-money valuation at the time that we go out and seek additional investment, which we have actually had discussions favorably at that number right now.
Yeah, so I believe our current valuation view is about 10 million. That's consistent with my view as well.
Okay. Last question due to time. Jeff H. wants to know, have you and are you now working with a joint venture with any major biotech companies?
Nothing formally, other than our, one of our science advisors being an employee at Moderna. He had to disclose to Moderna that he was working with us, and they approved his involvement. So they are at least aware, but we have no formal working relationships with anyone at the moment.
All right, gentlemen. Well, thank you so much for your time today. We certainly hope you come back and give us some updates in the future.
Excellent.
Thank you very much. Appreciate the time.
Okay, everyone, stay with us as we transition to the next presenter. We'll be right back.